Our mouths are incredibly busy places. They are home to billions of tiny living things called bacteria. This collection of microbes is known as the oral microbiome. For a long time, we have known that these bacteria play a big part in our health. Some are helpful, while others can cause problems like cavities or gum disease. But recently, scientists have found something completely new and unexpected hiding within this microscopic world. They have discovered enormous loops of DNA which they have named inocles. This discovery opens up a brand new chapter in our understanding of the life teeming inside us, suggesting there is still so much we have yet to learn about our own bodies and the microscopic partners we live with every single day. Imagine a bustling city, full of inhabitants going about their daily lives. That is what the inside of your mouth is like for bacteria. They have to deal with constantly changing conditions. Think about it. One moment they are washed with sugary drinks, the next with hot coffee. They also face attacks from our immune system and from other competing microbes. To survive in such a challenging environment, these bacteria need special tools. It turns out that inocles might be one of their most important survival kits. This recent finding is like discovering a secret library of survival guides that these bacteria have been using all along, hidden right under our noses, or rather, inside our mouths. The discovery of inocles is truly remarkable because of their sheer size. We are used to thinking of bacteria as having their main chromosome, a circular strand of DNA, and sometimes smaller DNA loops called plasmids. Plasmids are known to carry extra genes that can help bacteria, for instance, by giving them resistance to antibiotics. However, inocles are different. They are absolutely massive, sometimes even larger than the bacterium's entire main chromosome. Finding such a gigantic genetic structure that nobody had ever seen before is a major scientific event. It challenges our existing knowledge and forces us to reconsider how bacteria evolve and share information with one another in complex environments like our mouths. For decades, scientists have studied the bacteria in our mouths using various methods. They have cataloged many different species and have learned a great deal about their genetics. So, how could something as enormous as an inocle have been missed for so long? The answer lies in the technology we were using. Traditional methods for sequencing DNA involve breaking it up into very small pieces, reading those pieces, and then trying to stitch them back together with a computer. Because inocles are so large and have many repeating parts, this process simply did not work. The computer programs would get confused and discard these giant loops as errors, leaving them completely invisible to science until now. So what are these mysterious inocles? In simple terms, an inocle is a huge circular piece of DNA found inside certain types of bacteria, specifically those from the Streptococcus family, which are very common in the human mouth. Unlike the primary chromosome that contains all the essential genes for the bacterium to live, inocles are considered extra-chromosomal. This means they are not strictly necessary for the bacterium's basic survival under normal conditions. Instead, they appear to carry a collection of specialized genes. Think of it as a professional carrying a briefcase full of specialized tools for specific jobs, separate from the everyday items they need just to get by. The genes found on inocles seem to be all about helping the bacteria cope with stress and difficult situations. Life in the mouth is tough, as we've discussed. These bacteria need to handle sudden changes in temperature, acidity, and the presence of chemicals from our food and toothpaste. The genes on inocles provide the instructions for making proteins that can help the bacteria repair their own DNA when it gets damaged, or build stronger cell walls to protect themselves from attack. This makes them incredibly adaptable. It's a clever survival strategy, allowing the bacteria to thrive in a place where many other microbes would quickly perish, ensuring their continued presence in our oral ecosystem. One of the most fascinating functions of inocles is their role in something called competence. This is the ability of a bacterium to grab pieces of DNA from its surroundings and incorporate them into its own genome. Inocles contain genes that supercharge this ability. This means a bacterium with an inocle is much better at picking up new genetic tricks from its neighbors. For example, if a nearby bacterium dies and releases a gene for resisting a certain type of stress, the inocle-equipped bacterium can snatch it up. This allows for very rapid evolution and adaptation, making the bacterial community in your mouth a dynamic and resilient population that is constantly changing. The discovery itself was only possible because of a breakthrough in technology. Scientists started using a new technique called long-read sequencing. Unlike the old methods that chopped DNA into tiny fragments, this new approach can read very long, continuous stretches of DNA. When researchers from the Forsyth Institute and the University of California, San Diego, applied this method to bacteria from human saliva, they were stunned. They saw these enormous DNA sequences that had never been documented before. It was this new technological lens that finally allowed them to see the full picture and reveal the existence of inocles, solving a puzzle that had been unknowingly overlooked for many years. Perhaps the most astonishing part of this discovery is the unexpected connection between inocles and cancer. The researchers didn't just look at the bacteria, they also looked at the health of the people the bacteria came from. They compared the oral microbiomes of people with head and neck squamous cell carcinoma, a type of cancer, to those of healthy individuals. What they found was a striking pattern. People who were healthy and did not have cancer tended to have a much higher abundance of inocles in the bacteria in their mouths. This correlation was strong and suggested something very important was going on. This finding raises a tantalizing question. Could these giant DNA loops in our bacteria be protecting us from cancer? The answer is not yet clear, but there are some interesting ideas. One theory is that the bacteria carrying inocles are better at maintaining a healthy and stable environment in the mouth. By being more resilient and better at surviving, these good bacteria might outcompete other more harmful microbes that are known to cause chronic inflammation. Chronic inflammation is a major risk factor for developing many types of cancer, including head and neck cancers. So by keeping the troublemakers at bay, inocles might indirectly lower a person's cancer risk. Conversely, the study showed that individuals with head and neck cancer had significantly fewer inocles in their oral bacteria. This suggests that the absence of these protective DNA loops might create an environment where cancer is more likely to develop. Without the stabilizing influence of inocle-carrying bacteria, the oral microbiome could become unbalanced. This imbalance, sometimes called dysbiosis, might allow pro-inflammatory bacteria to flourish. The persistent inflammation they cause can damage human cells over time, leading to genetic mutations that can eventually result in the uncontrolled cell growth that we call cancer. It's a complex chain of events, starting with a tiny loop of DNA. It is very important to remember that this is a correlation, not yet proven causation. We cannot say for certain that having more inocles prevents cancer or that having fewer causes it. However, the link is strong enough to be a major focus for future research. This finding could be a significant step towards developing new ways to assess cancer risk. Imagine a future where a simple saliva test could tell you about the health of your oral microbiome and your potential risk for certain diseases. By analyzing the presence of inocles, doctors might be able to identify people who need closer monitoring long before any cancer actually develops. The discovery of inocles has opened up a whole new avenue of scientific inquiry. Researchers are now eager to dive deeper into understanding exactly how these giant DNA loops work. The immediate next step is to figure out the precise function of the many genes located on them. While we know they are related to stress response and DNA sharing, the specific jobs of most of these genes are still a mystery. Scientists will conduct laboratory experiments where they can add or remove inocles from bacteria to see exactly how it changes their behavior. This will help confirm if they truly make the bacteria more resilient and how they influence their environment. Another critical question is how inocles are passed between bacteria. We know that smaller plasmids can be transferred quite easily, but moving a piece of DNA that is larger than the cell's own chromosome presents a significant biological challenge. Understanding this transfer mechanism is key to knowing how these beneficial traits can spread throughout the bacterial community in our mouths. Do they have a special system for this transfer, or does it only happen under specific circumstances? Answering these questions will tell us a lot about the dynamics of our oral microbiome and how it maintains its stability and health over time. Beyond the mouth, scientists are now wondering if inocles, or similar giant DNA loops, exist in other parts of our body. The human gut, for instance, is home to trillions of bacteria living in another complex and challenging environment. It is entirely possible that similar survival structures are present there, also playing a crucial role in our health. Researchers will likely start using long-read sequencing on samples from different body sites to search for these hidden genetic elements. This could lead to a wave of new discoveries, expanding our understanding of the human microbiome as a whole and its intricate relationship with our well-being. Ultimately, this research could have a profound impact on everyone's health. If inocles are proven to be protective, we could see the development of new probiotics. These would not be just any bacteria, but specific strains of streptococcus that are known to carry inocles. Taking such a probiotic could help to fortify our oral microbiome, making it more robust and potentially lowering our risk of diseases linked to inflammation, including certain cancers. The journey to understand inocles has only just begun, but it highlights a beautiful and complex truth. Sometimes the biggest discoveries about our own health can be found in the smallest of our companions.